Topic: Cells, the basic unit of life. Aims of lesson. At the end of this lesson, you should be able to know the following: molecular makeup of cells, cell structure and function, roles of the following structures: cell wall, cell membrane, nucleus, and cytoplasm. Introduction. Revise organic and inorganic compounds that were covered in lessons 2, 3, and 4. Cells are mostly made of proteins, carbohydrates, lipids, nucleic acids, and water. Cells differ in size, shape, and structure in order to carry out specialized functions. Cells contain organelles with structures adapted to perform specific functions within the cell. Cells are very small, and it is through the development of microscopic techniques that we have been able to visualize and understand them. Concepts and skills. Below are definitions of some of the terms that you will study under this section. Use the glossary in your textbook to define these new terms/words. Note: at least 2 marks are awarded if you can define a term correctly in a test or examination. Diffusion, a movement of molecules from a region of higher concentration to a region of lower concentration. Chromatin network/chromosomes, long structures that consist of DNA and occur in the nucleus. Cellulose, a polysaccharide that is laid down between the fibrils of cell walls. Turgor pressure, the firmness in a plant cell due to the pressure of water in it. Cell wall, outer, non-living boundary of a plant cell. Phospholipids, type of lipids that form part of the cell membrane. Study the following information on cell structure and function. The roles of organelles. Microscopy. The microscope most commonly used in laboratory microscopy exercises is a simple light microscope. Use your textbook and study the functions of the different parts of the light microscope in preparation for practical work. Calculating magnification. Microscopes magnify an image using a lens found in the eyepiece, which is also known as the ocular lens. The image is further magnified by the objective lens. The magnification of a microscope is magnification power of the eyepiece x the power of the objective lens, e.g., if the magnification of the eyepiece is 4x and the objective lens 10x, then the total magnification is 4 times 10 equals 40. The specimen is therefore magnified 40 times. Measuring the size of cells. Cells are very small and their size is normally measured in micrometers, μm. The magnification of a cell or organelle is often indicated on a micrograph. A scale can also be indicated on the micrograph. Example: calculate the actual length of AB from the image shown in the micrograph given with the scale bar given below. Image: electron micrograph showing rough endoplasmic reticulum with a scale bar given. Measure the length of AB with your ruler, i.e. plus or minus 20 millimeters. Measure the length of the scale bar, i.e. plus or minus 5 millimeters. Use the following formula. Cells. The cell wall is a rigid, non-living layer that is found outside the cell membrane and surrounds the cell. A cell wall only occurs in plant cells. The cell wall consists of 3 parts, i.e. the primary cell wall, middle lamella, and the secondary cell wall. The primary cell wall occurs on the outside of the cell membrane and consists of cellulose. The middle lamella consists of pectin and occurs outside the primary cell wall. It connects the cell walls of adjacent plant cells. The secondary cell wall develops between the cell membrane and the primary cell wall in older cells. It consists of cellulose with lignin in between. Functions. The cell wall is a support structure that protects the living contents of the plant cell. It also gives rigidity to the plant cell. The cell wall is completely permeable to water and mineral salts. Cell membrane. The cell membrane forms the outer living boundary of the cytoplasm. All plant and animal cells have cell membranes. According to the fluid mosaic model, the cell membrane consists of a double layer of phospholipid molecules with large protein molecules embedded in the layer of phospholipids. The double layer of phospholipids is impermeable to water-soluble molecules such as glucose, amino acids, salts, and nucleic acids. Each phospholipid molecule consists of a head that attracts water, hydrophilic, and a tail that repels water, hydrophobic. Function. The cell membrane is selectively/differentially permeable and controls the movement of substances into and out of the cell. The following three processes are responsible for the movement of substances, e.g. water, gases, and salts into and out of a cell. Diffusion. The spontaneous movement of molecules of a liquid or gas from an area of high concentration to an area of low concentration. Diffusion is a passive process and no energy is required for this type of transport. Osmosis. The movement of water molecules from an area of high water potential to an area of low water potential through a selectively permeable membrane. Pure water has the highest water potential. Osmosis is a passive process and does not require energy. Plant cells use osmosis to absorb water from the soil and transport it to the leaves. Active transport. The movement of molecules from a low to a high concentration against the concentration gradient through membranes. Energy is needed for this type of transport. Nucleus. The nucleus consists of a double nuclear membrane with pores, the nucleoplasm, the nucleolus, and tangled threads known as the chromatin network. The chromatin network shortens and thickens to form chromosomes when a cell divides. Functions. The nucleus controls the activities of the cell. The chromosomes carry hereditary characteristics. Cytoplasm. Some of the cytoplasm is in a liquid form and some is in a jelly-like state. It contains dissolved nutrients and waste products. In eukaryotic cells, all the organelles are contained within the cytoplasm except the nucleolus, which is contained within the nucleus. Functions. The cytoplasm stores substances. Substances circulate through the movement of the cytoplasm. Know the meaning of instructional verbs in test and examination questions, e.g. Instructional verb. Meaning. Name. Give the name of something. Differentiate. Use differences to qualify between two or more categories. Tabulate. Draw a table and indicate the answers as direct pairs. Describe. State in sentences the main points of a process. Explain. Give your answer in a cause-effect or statement and reason sequence. Compare. Give similarities and differences between concepts. Answer the following questions. Question 1. 1. The diagrams below representing two cells with their organelles. 1.1 Identify parts D and I. 1.2 Which one of these diagrams, X or Y, represents a plant cell? 1.3 Give one visible reason for your answer in question 1.1. 1.4 What is the function of part C in cell X? 1.5 Name the organic substance that part K in cell Y mainly consists of. Question 2. 2. The diagram below represents an organelle, mitochondrion, found in plant and animal cells. 2.1 A scale showing 1 micrometer, 1 micron, is shown above the organelle. Measure the organelle between points A and B and use the scale to calculate the length of the organelle in microns. Show all your calculations. Question 3. 3. The diagrams below illustrate a process through a differentially permeable membrane. 3.1 What is meant by a differentially permeable membrane? 3.2 Describe what happens in Figure 2 and explain your answer by indicating in which direction the water moves and why. 3.3 Which physiological process is represented in Figure 2? 3.4 Describe the structure of the cell membrane according to the fluid mosaic model. Common errors made by learners in examinations. I unable to answer questions based on microscopy and diagrams of cell structure. Unable to answer questions based on experiments of diffusion and osmosis. Activities/assessment. Complete the questions given in this lesson. Consult your textbook, the internet, and other resources to find out more about the fluid mosaic model of the cytoplasm. Consolidation. Work through questions on microscopy and cell structure in past examination papers. Reflect on your learning and understanding of this topic and assess your progress. Values. By studying this section, you will develop your knowledge of key biological concepts, processes, systems, and theories. Education is the most powerful weapon you can use to change the world. Nelson Mandela.